Welcome friends for session 3 online session class 12th subject biology chapter number 2 sexual reproduction in plants. I am GR Chopde, PGT biology, JNV Sangli. So before this you can watch on YouTube session 1 and session 2 for chapter 2nd. So during this session, what are the points for discussion? Double fertilization, endosperm and embryo development, apomixis and polyembrony. Then finally summarize and you can solve the assignment. So before this we have seen stamen as a male reproductive organ, pistil is a female reproductive organ. In anther there is a microsporogenesis. In ovule there is a megasporogenesis. Then pollination takes place. And after pollination, there may be self-pollination or cross-pollination. Then there are certain outbreeding devices we have seen. Then mode of pollination we have discussed and pistil-pollen interactions. Now once the pollen deposited on the stigma, it germinate and produce after transfer the male gametes and cause a fertilization. So in angiosperm, double fertilization is the characteristics. Fertilization is first coined by a Starsberger. Now Sitchin coined the double fertilization. He discovered the double fertilization in plants. One of the male gamete moves towards the egg cell and fuses with its nucleus. Okay, that is egg nucleus. Okay, thus completing the syngamy. That result is formation of zygote. Then other male gamete move towards the two polar nuclei located in the central cell and fuses with them to produce triploid primary endosperm nucleus pen, and that is called triple fusion. So therefore in the same embryo sac two fusion takes place. One formation of zygote, one formation of triple fusion, that is endosperm. So therefore it is called as double fertilization. Here you can see, so the pollen tube germinate, it carry the male gamete. One of the male gamete will fuse with the egg cell, okay, that result in formation of zygote, and another male gamete move towards the two polar nuclei, and here it is result in formation of, that is PEN. You can see in another diagram here. See, these are polar nuclei and this is the egg cell. See, after fertilization, okay, that egg cell becomes zygote, diploid, and this polar nuclei become primary endosperm nucleus, that is triploid, 3N. And this is the only cell which has a 3N number of chromosomes. Zygote has 2N number of chromosomes, and remaining syncytids will degenerate and antipodal cells will degenerate. So this is the result of double fertilization. Okay. Now after fertilization, that is post fertilization, what will happen? Egg become zygote, central cell become endosperm, ovule becomes seed, integument become seed coat, ovary becomes fruit, and the zygote later on become embryo. Okay. Now first we'll see endosperm development, because endosperm should divide first before developing the embryo, because endosperm provide food to the embryo. So the primary endosperm cell, that is pen, divides repeatedly by mitosis and forms a triploid endosperm tissue. एंजियोस्पर्म का endosperm हमेशा triploid होता है, 3N. The cells of this tissue are filled with reserve food material and which are used for the nutrition of the developing embryo. So therefore endosperm development takes place first, then zygote embryo development. Okay, ये याद रखना है. Now types. So first one is free nuclear endosperm development. Here the pen undergoes successive nuclear division. यहाँ पे देखेंगे one nucleus is there. So this nuclei repeatedly divide and only nucleus are produced. After that the cell wall formation takes place from the periphery to the center. यहाँ पे देख सकते हैं. तो पहला न्यूक्लियर डिवीजन होगा, इसीलिए इट इज कॉल्ड फ्री न्यूक्लियर. और वंस इट इज कंप्लीटेड followed by cell wall formation. It is very common in cotton, maize, capsella या coconut. जो coconut का आप milk बोलते हो, that is a free nuclear endosperm. और फिर उसके बाद that coconut का अंदर का white color, that is called copra, COPRA. So that is cell wall formation. Second है that is cellular. Now see the difference. Every nuclear division is followed by cytokinesis. पहला nucleus then divide, 2 nuclei followed by 2 cell. Cell wall तैयार हो गया. So every nuclear division followed by cell wall. So इसीलिए it is called cellular. Best example blossom, datura, petunia, utricularia. या जो coconut का जो copra, COPRA. So ये cellular में आता है. So therefore coconut first show free nuclear and then show cellular. Okay. Now the third category है that is halobial. First mitosis followed by cytokinesis. Now यहाँ पे देख सकते हैं subsequent divisions are free nuclear. One nucleus is there, pen, it will divide. Okay, then there is a cell wall formation first. Then only the nuclear division takes place, nuclear division, and then slowly from here the cell wall formation takes place. So halobial, half. Okay. You can best see in Asphodylus and Eremoras. So for CBSE, just to read free nuclear or cellular, that is coconut example. For competitive exams like NEET, you have to know these 3 types. Now, endosperm may either be completely consumed by the developing embryo before seed maturation, or बेस्ट एग्जांपल पी, ग्राउंडनट बीन्स, वहां पे पूरा यूज होता है, सिर्फ कॉटिलिडन हो जाते हैं. और इट में परसिस्ट, ऐसा ही रह जाता है इन द मैच्योर सीड एंड बी यूज्ड ड्यूरिंग द सीड जर्मिनेशन. बेस्ट एग्जांपल कैस्टर एंड कोकोनट. सो द फर्स्ट कैटेगरी आर कॉल्ड नॉन एंडोस्पर्मिक सीड एंड द सेकंड कैटेगरीज आर कॉल्ड एंडोस्पर्मिक सीड्स. वो आगे भी आएगा. नाउ कम टू द एम्ब्रियो डेवलपमेंट. Develops at the micropylar end. Zygote elongates, पहला लंबा होगा, and divides to produce 2 unequal cells. The larger basal cell, it becomes suspensor and terminal cell. The basal cell divides repeatedly to produce a row of 4 to 8 cells, और उसको हम बोलेंगे suspensor. That will push the embryo towards the endosperm. The terminal cell undergo repeated mitosis division and form a cluster, a group of cells, and that is called proembryo. यानी embryo की शुरुआत होती है, उसको बोलेंगे proembryo. Now you can see here the zygote पहला enlarge हुआ, then unequal division. So this is smaller cell, this is larger cell. Okay, फिर ये ऐसा ही रह जाएगा. Again this will divide and become a suspensor, and the terminal cell will divide and become a proembryo. From proembryo it will become a globular embryo, या octet भी बोलेंगे. So then there is a differentiation. It will become a heart shape embryo. Suspensor will remain as it is, haustoria will remain as it is, and this heart shape structure will differentiated into radical part, here plumule part, which is covered by a cotyledons. So this is the mature dicot embryo. Okay. Now अभी ये corn या monocot seed, and here you can see the embryo. So this is entire endosperm and here is the embryo. So here a gram p, yeah, p, even you can take, this is there, if you take a cross section, cotyledons too, and inside will get the embryo. Okay, so dicot embryo structure, very very important. Diagram निकालेंगे. Consists of embryonal axis and 2 cotyledons. Portion of the embryonal axis above the level of cotyledons is called epi. Epi above cotyledons, above the cotyledons, which terminates with the plumule or stem tip या shoot tip. The cylindrical portion वो cylindrical होगा below the level of cotyledons, hypo below cotyledons, that terminates at the lower end of the radical or root tip. तो ऊपर का shoot tip epicotyle और नीचे का root tip, that is hypocotyle. Now you can see this in the diagram. See, this is seed coat, hilum, and there is a small opening called micropyle. So when you open this one, you will get a 2 cotyledon and in the embryo. If you see this young embryo, so here is a root cap, radical, and this cylindrical portion is called hypocotyle from here to here. And here is the plumule, from this part to this part is called epicotyle. Okay, plumule is surrounded by cotyledon, well protected. ये diagram निकालना है. Monocot embryo possess only one cotyledon, that is called scutellum. Cotyledon is called scutellum, that is situated towards the lateral side. Lateral में होगा. Lower end has the radical and root cap enclosed in undifferentiated sheath called coleorhiza. Rhiza root, coleorhiza. Epicotyle has a shoot, epi above, shoot apex and a few leaf primordia. यानी वहां से leaf आगे तैयार होंगे. Enclosed in a hollow polyar structure, that is coleoptile. एग्जाम में डिफरेंस आएगा, डिफरेंस बिटवीन कोलियोराइजा एंड कोलियोप्टाइल. ओके, तो ये लिख सकते हैं. सी दिस इज ए मेज. सो दिस इज 11th में आपने ऑलरेडी स्टडी किया है. सी कोलियोप्टाइल, देन प्लूमुल, या रेडिकल, दैट इज कोलियोराइजा, और ये एंडोस्पर्म में एलिरॉन लेयर है, सीड कोट एंड फ्रूट वन. ओके, ये लैटरल साइड में होगा. सो दिस इज ए स्कुटिलम. ओके, दैट इज कॉटिलिडन. नाउ ये डायग्राम अभी इंपॉर्टेंट है. दिस वन यू ड्रा. Again same, radical, root cap, coleorhiza. Here a epiblast भी आएगा. Shoot apex, these are leaf primordia या coleopter, and this is the scutellum. Okay, that will provide. So very very important, monocot embryo. Now, so these are different fruits. See that epicarp, mesocarp, endocarp. Which is the edible part in mango? That is mesocarp. But here edible part कौन सा है? Endocarp. ऐसा. Here you can see thalamus seeds here, and this thalamus is not from the ovary, it is thalamus part will become a fruit. तो ये है true fruit और ये है false fruit. ये भी false fruit है. Okay, that is achene type in strawberry. Fruits. The ovary wall is termed as a pericarp. Carpel, peri surrounding, pericarp. So it has three layers: epicarp uppermost, mesocarp middle, and endocarp inner, जिसके अंदर सीड होती है. Fruits may be true fruit developed from a real ovary. True fruit may be dry होगा. For example, you can take almond seed या almond fruit, और fleshy होगा. Orange ले सकते हैं, mango ले सकते हैं. Again, true fruits may be simple होगा, that is developed from single carpel. Aggregate group होगा या multiple carpel से तैयार होता है. Multiple fruit, that is multiple flower से तैयार होता है, यानी whole entire flower. Okay, so true may be simple, aggregate, multiple या dry और fleshy. You read any reference, ये पूरा details मिल जाएगा for competitive exams, but CBSE इतना काफी है. False, that is false fruit, fruit develop other than ovary part, maybe thalamus. Okay, ऐसा. now यहाँ पे देख सकते हैं अलग types of fruits आपको दिखाया गया है. Okay, so these are all aggregate fruits. And this is multiple fruit. Best example ये कौन सा? पाइनएप्पल. और यहाँ पे जो बेर्स वगैरह आप देखते हैं, कस्टर्ड एप्पल वगैरह आता है, एग्रीगेट फ्रूट्स. टोमेटो या मैंगो वगैरह जो आता है, बेरीज आती है या सिंपल फ्रूट्स. नाउ पार्थिनोकार्पिक फ्रूट. सी दिस वाटरमेलन एंड शिमला मिर्ची. फ्रूट्स डेवलप विदाउट फर्टिलाइजेशन. जब फर्टिलाइजेशन नहीं होगा, नो सीड. Okay, it was given first time discovered by Westwood in 1993. There are two types of parthenocarpic: vegetative, that is pear, and stimulative, that is grapes and banana. And by giving the hormones, growth hormones, you can make them to become parthenocarpic fruit. Tomato या pomes, fruits which do not have seed. So seedless fruits are generally developed from parthenocarpic process. Now seed, it is fertilized ovule या mature ovule. Seeds are formed inside the fruits, consist of seed coat. Outermost is testa, innermost is tegmen, और ये इंटुगमेंट से तैयार होते हैं. Cotyledons या cotyledon and an embryo axis. Mature seed may be non-albuminous, that is, or albuminous, non-endospermic या endospermic. Perisperm, कुछ सीड्स में रेसिडिवल, persistent, nucleus, जहां से मेगास्पोर डेवलप हुआ था, रिमेनिंग सेल ऐसे ही रह जाते, दैट इज कॉल्ड एज पेरिस्पर्म. पेरी यानी आउटर मोस्ट. So that you can see best in black pepper. Okay, black pepper. Seed dormancy. The metabolic activity of the embryo slow down. Yeah, or embryo may enter a state of inactivity. It cannot germinate. But if the favorable conditions are available, adequate moisture is there, oxygen is there, normal temperature is there, they germinate. So they overcome seed dormancy. And seed dormancy is very, very important, okay, for plant growth, even storage of food. Now, seed dispersion. 8th class में आपने पढ़ा है, carried away by wind, water or animals. Winged seeds जैसे कि drumstick और maple है. Light seeds of grassy or hairy seeds like oak or sunflower. Spongy or fibrous seed, that is coconut, transferred by water. Spiny seeds with hooks, xanthium and urena, that is transferred by animals. Okay. Sudden jerk भी होता है castor और blossom में. So ये just revise है for your competitive exams. Here आप देख सकते हैं by wind ragwort है या winged seed, यहाँ पे drumstick है या इधर sudden jerk वगैरह होता है. By animals ऐसे hook like structure होते हैं या when the animals eat these black bears, okay, through that digestive system excrete वो ऐसे ही चला जाता है. Now, so there are certain plants like Lupinus archaticus, arctic tundra, germinated after 10,000 years of dormancy. And dormancy period इतना होता है. 11th you have studied what are the causes of seed dormancy. Phoenix dactylifera, king heroids, palace near the Dead Sea मिला है and it is 2000 years old seed. यानी इतना उनका dormancy period है. You can see a 1 mark के लिए आएगा. You can see this a Lupinus archaticus and this is the second group, that is Phoenix dactylifera. Okay. Now, what are the advantages? Seed. It stores sufficient food reserves. Young seedlings are nourished. Protection to the young embryo. Generate new genetic combination, क्योंकि male और female का combination. Storage of seeds which can be used as a food या future generation. Okay, ऐसा. Now, what is apomixis and polyembryony? Produce seeds without fertilization. That is called apomixis. See difference between parthenocarpy and apomixis. Parthenocarpy, fruit develop without fertilization. Here, seed develop without fertilization. समझ में आ गया? Very important. So, how the diploid egg cell is formed without reduction division? We have studied reduction division होता है, haploid egg cell तैयार होता है. But here, okay, apomixis, produce seeds without fertilization. Diploid egg cell is formed without reduction division and develop into the embryo without fertilization. हमने देखा है, okay, without ये. So this is how development takes place. Citrus and mango are the best examples. Citrus and mango. Nuclear cells also develop into embryo, okay, in the embryo sac and they become embryo. तो इसीलिए there are more than one embryo in one embryo sac. So that condition is called polyembryony. Presence of more than one embryo in a seed is called polyembryony. Okay, very, very important. Now, what are the advantages भी उधर आपको पूछेंगे to see that how the seeds will get germinated. Okay, ये wheat का seed है. Once the seed produce, so they will germinate. See, पहला root radical will develop, then plumule will develop, shoot develop. Okay, अभी ये epicotyle और hypocotyle we have studied. Above the और below 11th standard ये ऐसा है. First था, दूसरा और ये था. Okay, beautiful seed germination. And then the plantlet will become, and this plantlet will grow, again grow for the 3, just a juvenile phase, reproductive phase and senescent phase. During reproductive phase, it again flowers, again male gamete, female gamete, double fertilization, seed, fruit, seed dispersal. So the life cycle will repeat. So this is a means. Okay. dicot or monocot. I have shown both. Okay. So this is about what? A second chapter, reproduction in plants. So you watch all those 3 videos and solve the question answer given at the end. So in next, I will come with certain questions. Thank you for joining my session.